Welcome back, everyone. Next up, we have Bioxytran. It trades on the OTC under the symbol BIXT. It's a clinical stage pharmaceutical company developing platform technologies in the fields of glycovirology, hypoxia, and degenerative diseases to eliminate viruses and prolong lifespan using carbohydrate drug design. Please welcome its Executive Vice President of Business Development. We have Sheikh. Welcome, Mike. How are you doing today?
Oh, doing great. Can you see my slides?
I believe so pretty soon, but we look forward to your presentation.
All right. Anna, thank you for that great introduction. My name's Sheikh. I'm going to deliver your presentation today. I'm the EVP of Bioxytran, Inc. But first, what I want to do is I want to thank Emerging Growth for allowing us to present at this conference. The reason why is I believe we're the first ever Caveat Emptor stock to present at Emerging Growth. And honestly, I think that speaks volumes to be able to present on this stage, being in the status that we are. Look, so this presentation is going to be really focused on the science, so you can see the actual incredible potential of our platform technologies. So with that, I want to say we are Caveat Emptor, which means buyer beware, which means don't buy our stock yet. Okay. Hopefully in the future, we'll get that flag removed. We don't know when, we don't know if. So those are all the disclaimers right now. All right, let's get started. So we have some forward-looking statements here, and you can read through that. So let's hop right into it. Our mission statement. You're going to see that we're a clinical stage pharmaceutical company. It means we're beyond the research side of things. We're into a clinical development, and we're looking at the fields of glycovirology, hypoxia, and degenerative diseases. And our goal is really to eliminate viruses and prolong lifespan using carbohydrate drug design. You'll understand what that means. And hopefully the pictures down below, you'll see glycovirology. It's clearly with viruses, hypoxia. It deals with oxygenation and degenerative diseases, stuff when you get old. We broke our technology down into two buckets, two columns, if you would. Look, you'll see on the left side is our glycovirology. On the right side is our hypoxia and degenerative diseases. So on the left side, you'll see the glycovirology. One of our biggest case studies is in COVID-19. And then you'll see there's influenza, other virological diseases. And then it goes into, possibility of long haulers and stuff and such. So on the right side, you'll see that, we're focused on ischemia, which is the disease associated with ischemia, stroke, Alzheimer's, dementia, traumatic brain injury. And then you'll see underneath that, there's some platform overlap. This platform overlap, quite frankly, could be some combination of uses or dual uses in one case or the other. All right. Now, our main experimental drug candidate is prolactin M. You'll see that in the lower left. It's a licensed technology that we're looking to license the technology that targets COVID-19 in mild to moderate cases. OxySense, we're not going to really delve into that too much because what it is more of a companion. It's an actual device, FDA-approved device, but we're going to use it in a companion diagnostic role with BIXT 25 to help us get through the indications of stroke. So glycovirology, I'm throwing out a term that hasn't been used before. So what does that mean? Well, we're basically using carbohydrate drugs to defeat viruses. And that's what the study of glycovirology is all about. So with that, let's get into our first case study. COVID-19, it's still out there. And what happens is these variants are really complicating the vaccine strategy. We don't have herd immunity yet. We don't think we're going to get it. The vaccinations, I mean, there's some people who want it, some people who don't want it, and there's some people that want it and can't get it. I mean, so we have an issue with the vaccination strategy, and new mutations are all around us, and then people are still dying right now. I mean, if you punch up COVID cases in the United States, you'll still see death. So there's we still have the at-risk population dying. And finally, there's asymptomatic spread. There is just an incredible lack of testing out there to control the community spread, and it's still going to be with us until we do something about it. And it doesn't look like we're doing anything about it. So there's risk with vaccinations, but here's the bottom line. If herd immunity is unattainable, therapeutic treatments are the only fallback position. And that's where we come into play. Our platform technology is a galectin antagonist platform. All right. What makes us so special is that it's versatile, which means that it doesn't matter what mutation it is, whether it's Omicron or Delta, doesn't matter. It's tested. We do have an academic study that was completed that you'll see that right below it is no toxicity, reduction of viral load to undetectable levels. We don't expect any limitations. That means if you're young, if you're old, if you're pregnant, you're vaccinated, unvaccinated, it doesn't matter. We're efficient. That means we can quickly eliminate the contagion. That's what efficiency means. So we want to be the first line of defense against all mutations of coronaviruses. And you'll see down below there in the lower right-hand corner, we have submitted an IND. We believe our technology is an end-to-end solution. You'll see at the top in red, we have prolactin M, I, A, and F. The M is the oral version, and that's where we're focused right now. The intravenous is coming soon. I can't say when, but it's soon. And you'll see that the intravenous will be the same formulation, but used for different indications. This slide, let's focus in on the, you've probably seen this slide, but let's focus in on the red line. You see the viral curve goes up, you get symptomatic, and then it goes down. What you want it to do is go down to nothing as quick as possible. But we see with these other stages of disease, it still hangs around. And then finally, you'll see we believe that our technology could have an impact on fibrosis and long haulers. Here are some actual clinical results, clinical trial results from our academic study. You'll see in the graph on the left that we have a bunch of lines going down. That's good. That represents the viral load. And you want the viral load to go down and dive into that red line as quick as possible. Now, that red line represents 50 copies per microliter. That in HIV, for example, that represents an undetectable level of virus. All right, so that's what you want to see happen. Our data's on the right, and then you have the bar graph on the lower right-hand corner, and what you'll see is day 1, day 3, day 5, day 7. Look what happens at day 3. Look at how close those patients got to the undetectable viral load level. That's the story right there. For comparison purposes, there's a smooth line that you'll see there. That's the upper blue line, and it curves. That represents a historical control. Our source is right below there. It assumes a whole bunch of different things, but the bottom line, that is the trajectory of the viral load. You will see our trajectory of the viral load. It's within it. It reaches the red line faster. That's noticeable. Again, I point you to the footnote down below. It is a peer-reviewed trial, so you can take a look at it. This is also from that actual academic study. We found that the galectin antagonist treatment results in CoV-2 spike-specific antibody immunity. What does that mean? That means if you get infected, it helps you develop antibodies quickly to prevent another infection. So you get long-term protection from getting infected. What's happening in there is that the innate immune system, which are basically the first responders of your immune system, they go in there and they've got nothing to do because our galectin antagonist comes in there and takes care of business. And then the adaptive immunity, they are behind the scenes clearing up the virus and creating these antibodies in the background. And that's what leads to a robust response and prevents you against future infection. This is an illustration of our theoretical mechanism of action, what we are in the process of proving. You'll see in the first part of the picture, the virus enters the cell freely, no issues. When we introduce the galectin antagonist, do you see how it kind of globs on to the spike proteins? Basically, it attaches there and prevents viral entry. Now, what's interesting is that the liver sees the carbohydrate galectin antagonist and wants to filter it out of the blood. And that's what we believe the mechanism of action and what our research is hopefully in the right alignment to prove. Here is our peer-reviewed journal article, again, footnoted at the bottom. Let's get into the proposed clinical design. Please understand, this is proposed. It's subject to change. Right now, we're looking at 408 participants. We want to do a double-blind randomized controlled trial. We're looking for the change in seropositivity at day 14. What does that mean? We're looking for basically PCR negative. And broad inclusion criteria, it doesn't matter your vaccination status. It's irrelevant. This is our development timeline. You'll see we have prolactin M, I, A, and F. Let's focus in on prolactin M, which is an oral treatment. You'll see there we're going after COVID-19 first. That's our first case study, and then influenza and other virological diseases eventually. Where are we at? Well, we've submitted an IND. And you'll see the next step is to figure out what phase of development that we want to go into. Now, a lot of people are under the impression that the FDA directs that. That's not necessarily the case. It's in consultation with the company and the FDA that we figure out what phase of trial that we want to enter. So that's why you'll see it's up in the air there. The next thing is prolactin I. And again, our case study, the first case study we're going to go after is COVID-19 in severe disease. And you'll see we have not yet submitted an IND. It's in the preclinical stage, but we hope to get into the IND stage shortly. I can't give you any time horizons, but that's the next step. Our focus again is on prolactin M. Now, where do we fit in the whole universes of therapeutic approaches for COVID-19? For example, you'll have antivirals. You'll have immune modulatory. Now, the antivirals, we all heard of them, Paxlovid, Molnupiravir, those are the ones under current EUA. Immune modulatory, you're looking at drugs like tocilizumab, the IL-6 inhibitors. Vaccines, we all know about Molnupiravir, Pfizer, and Johnson & Johnson vaccines. And regenerative medicine, that really revolves around stem cells. And then oxygenation, we all know about the ventilators to help people. Those are all therapeutic approaches to help people get better. Now, our prolactin technology fits in the center of this all. Okay, we're unique galectin antagonists. It could be via oral or IV. It's not a vaccine and it's not really an antiviral, but maybe we're an entry inhibitor. So our goal is to prevent the virus from getting into the cell. We think it's better. We think it's possibly a better way to go than allowing the virus in and then messing up with its mechanism inside the cell. This is the competitive landscape. You're looking at molnupiravir, you're looking at Paxlovid. You'll see that those are EUAs out there. And then some upcoming ones that are of note are Tolavir and Totos Medical. I think they were the presentation before us. And then Tempo and Adamis. Again, the real thing that I want to get out of this slide is governments are the end users with COVID-19 therapeutics. Big pharma involvement really isn't necessary. So I want to drill that home when you think that little pharma, like us, can't compete, at least in COVID-19. Perhaps the other indications, you'll see that our strategy is we do want to partner up. Now we're going to get into our other platform technology, BIXT25. A little background. This is what the company was built on. Hypoxia deals with oxygenating tissues, and degenerative diseases, we all know what those are. And let's get into one of the top issues with hypoxia, and it's a degenerative disease, both. And that's the stroke epidemic. Strokes. Look down there at the bottom, 12.2 at the bottom of the chart, 12.2 million people get strokes. Look at the cost, $500 billion. Actually, that cost is probably really low. We did conservative estimates on this. So you have indirect and direct costs. The indirect costs are basically caretakers after you have the stroke. So wouldn't it be nice to get to people, treat people early, and prevent all the damage that happens? And that brings me to my next slide, which talks about the golden hour dilemma. When you get a stroke, your brain is essentially holding its breath. It's waiting to establish that oxygen connection, that oxygen bridge again. So look down at the little bullets the little circles at the bottom. First, you get the onset of symptoms. That's where you pick up the phone and you call 911. 15 minutes later, the ambulance is going to arrive. Then they're going to go into ER. That's the next one. And then once you're at the hospital, you're waiting for imaging. You cannot get any treatment for stroke until you figure out what's an ischemic or hemorrhagic stroke. If it's ischemic, means you have a blood clot. If it's hemorrhagic, it means the blood vessel burst. That's simple. And then finally, you can get the treatment, the blockage is removed. Now, the time to needle is 2.5 hours. That whole process there is 2.5 hours. What happens during that 2.5 hours is your brain was holding its breath. It aged 9 years. So what if we could do something about that? What if we could develop an oxygen bridge? Now, there's a graphic there that shows our molecule, BIXT25. It's an illustration, and that illustration is nowhere near to scale. Our molecule is 5,000 times smaller than a red blood cell. Those little dots there don't represent that. So understand it's much smaller, but I wouldn't be able to show you what's going on. So the blue passes through the plasma to the other side and continues to oxygenate your blood, because despite a clot, you still have plasma that's flowing through. We believe that in theory, this is going to work in ischemic and hemorrhagic stroke. And look at this, you can get to it in 3 minutes. What does that mean? A paramedic arrives, they see the person having a stroke, they put an IV in them, and then your oxygen bridge is established, or hopefully established, with this. That's the idea and the concept behind our solution. So let's get into how it works. How does this thing work? So an erythrocyte is basically a blood cell. Blood cell has lots of hemoglobin. And then there's the heme of the hemoglobin, right? So, and the heme is what actually carries the molecule, and it does a really good job of it. And whether it's in a human or another mammal, we all use the same mechanism. The molecule is going to be delivered as an IV solution. This isn't a pill that you can take. It has to be IV. You have to get it into the vasculature, means your arteries and blood. And what it does is it goes around, it picks up an oxygen molecule, delivers it, does it again and again and again until it wears off. It's not immunogenic, which means your immune system doesn't see it. And we don't have any compatibility issues with blood type. How do we make it? Well, you start with the core blood cells. You extract the hemoglobin protein, and you end up with heme. Then you purify, cross-link it, extract the heme, and reattach a polymer to it. Long story short, it's a very complicated process that I'm simplifying. And then you end up with a little IV bag solution like that hopefully the paramedic can administer at the time of stroke. Now, I don't want to mislead people. That's the ultimate goal of where we're going to go. I don't think we're going to start on those first-line type patients. But I just want to give you an idea. That's the potential of this. We had some really incredible proof of concept in animals. And let's focus in on the middle right there. So what happened is you see a rat there, and his neck is opened up, and then you'll see a little clamp. Well, what happens when you put a clamp on it? Just look at the picture on the right. You end up with, in the upper right corner, you end up with the brain is got white there. Well, that's because there's no blood flow. The pink section has blood flow to that hemisphere of the brain. So what happens is you have a rat that looks like that, and then you introduce the molecule, similar chemistry to BIXT25, and what we saw is what's in the lower right hand of that picture, is we saw restoration of oxygenation, not blood flow, oxygenation to the brain. Now we advanced this a little bit more in animals, and you'll see in the upper right, it's highlighted in light blue. There was no toxicity replacing 90% of the blood in dogs with similar chemistry to BXT25. So there was a test done where we put a pick in the dog's leg. We took out 90% of the blood. I want you to think about taking about 90% of your blood, how you'd feel. So we took out the blood while we replaced it with something similar to BXT25. And in that model, the animal is supposed to die within 50% of the animal is supposed to die within 2 hours. Well, if you want to read the study, it's down there at the lower left. You can go to it. There's a link. I'll tease a spoiler alert. They all lived. They all lived and recovered. So that was an incredible finding. Right now in stroke, you have limited treatment options, and read what's in bold. There are no drugs available to deliver oxygen to the brain right now. The only treatment is the RTPA, and what you'll find out is that it's really the only treatment there is. The other treatments listed down there are essentially drugs that redistribute the oxygenation within the brain with the hopes that it would redistribute it toward the more affected areas. How do we measure tissue oxygen level? Something called OxySense. It's a FDA-approved companion diagnostic. It measures oxygen delivery to the brain or organs in real time. You'll see there's a couple parameters in there in the middle chart. and looks like an EKG. That's how we anticipate it, or that's how it works. And it develops a score to the right, which is called the tissue metabolic score or brain metabolic score. What's really interesting, and I don't want to get into this, but I'm kind of intrigued by this whole thing, is the potential here is eventually take something like this and miniaturize it and have it on like a watch or something on your skin or something. There's some really cool technology here, but that's not what we're using it for. We're using it as a companion diagnostic. We're using it to help us get the indication of stroke approved. So that's our only interest in it right now. But some cool technology. Here's our pipeline. Look at all the indications that we hope to go after. Now, you'll notice it's all in discovery phase. Nothing has reached preclinical status. Why? We need funding for it. Let's get into a business development strategy. We got prolactin M, we got prolactin I, F, A. Our focus is really on prolactin M. So let's just start and end it there. So right now, we're past the preclinical development, and we're trying to determine what phase of clinical trial that we're going to be in, and we're doing that right now. And that's in essence our strategy. We have intellectual property, 2 international patents pending. And then let's focus on the business development strategy. Remember what I said earlier. We want to collaborate with qualified partners, or we want to out-license the big pharma. When it comes to the COVID-19 strategy, clearly the governments are in play there, and that's who you deal with. Now let's look at who we would do business, who we would potentially partner with. Okay. What's the pool of partners? What I did was I took the therapeutic areas, virology, neuroscience, oncology, cardiovascular, it's right there on the top where it says therapeutic areas. And then what we did, so we start with that, and then let's look at big pharma. We looked at big pharma, and we looked at their pipeline, and we looked at all the different classifications of indications, and look at how many, our drugs hit all these points. We'd be an ideal platform drug for many big pharmas. Now we're going to get into use of proceeds. Now, quite frankly, this is taken almost cut and paste directly from our S1. So you're free to look at our S1. But let me just boil this down to, you'll see prolactin M, you'll see prolactin I, you'll see prolactin F, and then you'll see the future rounds. We hope to do prolactin A and then BIXT 25. So again, take a look at our S1. That's a really good source. But this is just an overview of what we're looking to do. Now let's talk about the team. In biotech, the team is quite important. Here we have Dr. David Platt. He's arguably the grandfather of galectins, and he's a carbohydrate chemistry expert. He's founded 4 public traded companies. 3 of them went on to achieve NASDAQ listing, raised money, created shareholder value, and he's led the development of 4 drugs. So we have an all-star at the top of our team in terms of drug development. Ola Soderquist, he's our CFO. I can't say he's very physically responsible and keeps us on track. You'll see that we've done a lot with very little. And then myself, I've been around life science a long time. I've been a broker and research analyst. So presenting to the investment community is something that's my skill set. And then Veronica is the one that keeps us on our timelines and milestones. There's our board of directors. We have some independent directors. Keep that in mind for potential uplisting. And then our advisory board. We have Abraham Majewski. He is the core behind our BIXT 25 platform technology, and Dr. Sigamani and Dr. Thomas Cuddy, or I'm not Dr. Cuddy, they're behind our Glycovirology platform. And that concludes our presentation. I'm ready for Q&A.
Thank you, Mike. Okay, let's jump right in. Frank Riley asks, what makes yours different from the other inhibitors, galactin inhibitors under development?
You know, it's really, I can say it in one word, chemistry. You have 15 different galactins out there, and which one are we going to choose to bind to? You know, do we want to bind to one? Do we want to bind to multiple? And then you have to consider the method of administration. Do you want to use oral, IV, or inhaled? So that differentiates all the galactins that are currently under development. So what makes us different? Well, we're oral, we're IV, we're not inhaled. So that's the only thing that makes us different. So others are more specified. I'd say that we're more general.
Okay. Lorena H asks, how much money has been used to develop this technology?
You know, a lot is the real answer. You got to remember, there's 2 platform technologies inside this company, not one. And if you put a gun to my head and say, I'd estimate you're probably looking at around 10 million plus went into the development of these technologies. There's a lot behind it, and I'm going way on the conservative side.
Gerald Harper asks, how big is your Glycovirology platform?
You know, the Glycovirology platform is really is a function of, for right now, it's a function of the size of the vaccination market. You see, right now you have 2 treatment choices. Look, do I want to beef up the antibody response, or do I want to use an antiviral like Paxlovid or molnupiravir to target the viral load? So it really comes down to strategy. Do we really think it's effective to vaccinate the world, or perhaps use a pill to prevent someone from dying? So it's really more a question of economics. Which is going to be more effective? And you know, look, we've seen vaccines; they have challenges left and right, fighting these new variants that are coming up. So I think that's how big we are. We're about as big as the vaccine market, our platform technology.
Interesting times. All right. Dan Bailey asks, you call this, excuse me, you call this a platform technology. How many viruses do you think you can ultimately target?
You know, look, glycovirology, this is a brand new science. You know, it's analogous to when Jenner developed the smallpox vaccine. That one vaccine led to how many other vaccines? I mean, you have vaccine, you know, the major ones, measles, mumps, rubella, influenza. Look, there's thousands of viruses out there, and we quite frankly don't know what we don't know yet. This is the beginning of a new field of research. So that's why this is very exciting. So, you know, you saw in my slide deck, we're going to go after influenza, but there's nothing to say we can't go after even the common cold. So that's how big this technology is. There you have it.
Don Allison asks, what makes you think you can compete against Big Pharma?
You know, Big Pharma isn't necessarily our competition. I mean, we're small pharma, but they're not necessarily our competition. Actually, go back to my business model. Our business model is basically to work with Big Pharma and license the technology. In order to do this, what do we need? We need a data set that they can wrap their hands around and sink their teeth into, which means we have to complete larger clinical trials. We have to prove it to Big Pharma that we're worthy, basically. Now, COVID-19, that is an entirely different subject. We've seen clearly that the governments are going to go directly to the pharmaceutical makers and buy the product. So in that sense, we do not need the Big Pharma distribution network we don't need the Big Pharma distribution network to distribute our drug.
Lorena H asks, do you think other galactin inhibitors like belapectin or GB0139 will work in COVID-19?
You know, these molecules are all galectin 3 inhibitors. So in theory, it clearly supports the idea of efficacy in COVID-19. Now, GB0139, that's a different story. You know, they actually had clinical trial results, and they were quite good in ARDS, you know, as it relates to COVID-19. And I believe there was a mortality benefit. I'd have to check, but you know, there's definitely some indications there. Now, I don't believe there's any studies on belapectin were done, or none that I know of, so I can't really speak to that one. But I don't know. Hopefully that answers the question.
It's okay. Jeanette Harrington asks, she says, I noticed your drug seems to attach to the spike protein. Do you think it can get rid of viral persistence associated with long COVID?
Now, if you're asking if the drug could work in long COVID, the real answer is I don't know. Now, if you want me to speculate on the theory of viral persistence, then yes, it's a possible theory that a galectin inhibitor could clear the virus from these viral reservoirs and possibly do it quite quickly. Now, the latest preprint goes into that we have out there, and if you follow me on Twitter, it's in my Twitter feed. It details how well our molecule binds to the galectin 3, and quite frankly, there's a lot of evidence out there. There's probably at least over 30 journal articles on the galectin 3, for example, and COVID-19 out there that show that galectins do go and bind to the spike proteins and on the galectin fold rather. So in theory, galectin 3 inhibitors could bind to the site and clear the virus. And if it's viral persistence and you take away the virus, you know, you possibly restore the immune system to normal function. So okay. there you go for COVID, long COVID possibilities.
Something's rustling in your audio. I'm not sure what that is. Last question for you from Kat Reeves. Do you think your galactin blockers can be used prophylactically?
You know, we've seen anecdotal evidence that this technology could basically prevent sickness. So the challenge is proving this. You would have to do a very large clinical trial to prove this. So I think that's ultimately what the issue is being able to prove that. So yes, it's possible, it's very likely, but it's unlikely that we're going to take the development pathway to prove this as a prophylactic use. So it's probably a pass.
Okay. We have a few more questions rolling in for you. We've got one from Michael Glorioso. He asks if BXT25 can pass through a clot in the brain, can't it pass through a clot in the heart for immediate oxygenation?
You know, that's a really good observation. The idea is, yes, I mean, that's the theory behind it. So could it be used in heart attacks? The theory definitely supports that for sure.
Mike, would you have any closing remarks you want to make?
Yeah, you know, I want to say that it's a real honor to present our company. This is the first presentation. Again, we're caveat emptor status, so obviously don't buy our stock until the caveat emptor flag is removed, if it is in fact removed. Because there can be no assurances of that. But we hope, I really hope that everyone see the potential of the galectin science. I've been studying galectin science for possibly 20 years. We have an all-star on our team, Dr. David Platt. He really knows what he's doing. And when you look at a biotechnology company, you really need to have a strong team that understands the clinical trial development process, understands what they can and cannot do, and is a steward of your funds. Again, we're focused on commercialization, not necessarily research. And the research has been done. So I really thank you for letting me present at this conference. It's been an honor.
Great presentation. Yes, the research has been done. Please join us again, Mike, with some future updates. We look forward to hearing from you again. All right, everyone, stay with us. We'll be right back with our next presenter.